Since the dawn of humanity, man has searched for the meaning of life. This simple question has long been a challenge for scientists and philosophers alike. Without getting into the complexity of the definition itself, today my endeavor is to present a new paradigm related to the living systems, characterized by the ability to organize, grow, adapt and reproduce. The new paradigm that I propose today adds a new dimension to the basis of life. While adaptation and evolution have long been used in scientific and philosophical literature, the mechanisms that led to the adaptation and evolution are often attributed to chance. My attempt today is to present a better than chance explanation for this fundamental attribute of life. Ever since the discovery of human genome, information is thought of as the basis of all life forms and near life forms like viruses, determining to a large extent life's ability to perform vital functions like selection of food, breaking up of food molecules into appropriate parts, selection of those parts and their assimilation. However, the ability to adapt and evolve is only partly explained by the information alone. There has to be something else other than information that governs the adaptation and ultimately leads to evolution over the longer term. I call that something as learning. In short, I define learning as the ability to adapt the function to the environment. At EPIGENOME, we hypothesize information and learning as the basis of life. The symbols of our information are 4 nucleobases, which contain know-how about the formation of proteins that enable us to be functional. Why is information only based on 4 nucleobases and not on others? Why are there only 20 amino acids and not more? All these are valid questions for another day. Today, however, we plan to delve deeper into learning, which is scarcely defined, much less explained in the context of life. The information that defines you is all encapsulated in 715 MB. Information that defines me is also encapsulated in 715 MB. While there may be minor differences between your information storage and my information storage, this alone is not what defines you and me. The information is categorized as exons or coded DNA, introns, non-coded DNA within genes, and intergenic non-coding regions. Non-coding DNA has so far not been properly understood. Perhaps it is crucial to learning, as the nature of learning has also not been understood so far. So why is learning important? I ask you just this one question. Someone said, little knowledge is a dangerous thing. Complete information in the absence of learning is deadly, is all I would say today. The knowledge of which information to use, when and how, comes through learning. Learning reflects in ourselves through epigenetics, literally meaning beyond genetics. Epigenetics determines in what manner we function in life via proteins, the key molecules through which functions are discharged. The formation of proteins in the human cells is controlled by epigenetics. There are 3 types of epigenetic changes which impact the gene expression or protein formation the most, and we will briefly talk about each of this. The first one is DNA methylation, wherein 2 specific nucleobases, adenine and cytosine, are methylated into 3 modified forms. The second one is histone modification, which impacts gene expressions by making parts of DNA inaccessible. The third is any RNA molecule that does not translate into protein is non-coding RNA. Many of the newly identified non-coding RNAs, initially referred to as junk RNAs, are increasingly being found to be associated with gene activity and expression. Which gene gets expressed and when is what determines you and me. Epigenetics is the determinant of this expression. Some of the proteins work as transcription factors which determine how much of the information will be used and in what sequence. By controlling expression of genes that make proteins called transcription factors, epigenetics also controls the rate at which information is acted on. Sometimes a small signal can also have multiple level cascading effect, which ultimately results in very large expression of certain proteins. A single methylation at one base pair can become either highly beneficial or detrimental to our survival through such cascading effects. Having touched on the nature of learning and the way learning is expressed in human cell, I will now outline attributes related to adaptation to environment, which are not explained by information alone, specifically our ability to respond to the same environment differently at different times. Our ability to adapt to the environment is characterized both in the way we learn a new skill and in building up strength or stamina. Any extreme change in environment can be deadly, but carefully calibrated change can lead to strong learning. In an experiment conducted by Kurt Richter in 1950s, he put similar rats in a bucket full of water and rescued them just before they are about to drown. Next time when the rats were placed in water, they outdid their last effort multifold. If a rat was about to drown in 2 minutes in the first attempt, next time it was still swimming after 60 hours. While Kurt outlined hope as the basis of such progression, I feel it was learning from the first effort that allowed the rats to keep swimming. If they had simply seen other rats being saved from drowning, which means the hope was still there but not first-hand learning, these rats would not have survived hours in the water. It is important for us to understand that learning is integral to all life forms and all kinds of cells. It is not the only prerogative of the brain. Learning as we know today is only defined in the form of mental understanding. What we are presenting today is a framework for organic learning which will redefine the way we look at all the past knowledge and research findings. For instance, the Kurt experiment on rats assumes a whole new explanation in the presence of learning. We can perhaps check rats' DNA methylation after such an experiment to check how is learning chemically expressed in the cells. In a recent experiment on a unicellular bacteria, E. coli, strong evidence of associative learning was discovered. I also feel we need further research to determine whether evolution comes by chance or via learning. Chance may not have led to proliferation of so many life forms. Learning further resolves the paradox of the plankton, wherein a limited range of resources support an unexpectedly wide range of plankton species. We have briefly defined the information and the nature of learning so far. We have further covered how learning is embedded in all life forms and is not exclusive to the brain. We have also outlined how learning contributes to adaptation to environment and could have more likely led to evolution of diverse life forms rather than just natural selection. In the next few minutes, I will build a model around learning that can be used as the foundation for targeting improvement in human health. The conceptual framework of artificial intelligence and machine learning can be used to develop the framework for the learning model. In the learning model of life, we see DNA as the training data for every cell. Every species having broadly similar data leads to similar physiology. While each of the cell type in a human body has identical training data, the way the cells read the data determines which type of cell it is. Specific cells in the human body, which are part of the immune system, have a mechanism to scan through the training data of any cell, and any differences would result in the body recognizing that as a foreign object and thus rejecting it. The tissue rejection in organ transplant is an example of this. The task of the immune system is to maintain whole body integrity, which needs the same set of training data. In identical twins, because of the identical nature of the training data, chances of tissue rejection are remote in case of an organ transplant from such twin to the other. Such cases are well documented in medical literature. Even with similar genotypes, phenotypes can differ widely based on interaction of the model with environment, like skin color, eye color, and so on. Every cell is continuously learning during its life to form a phenotype that would have different levels of expression of the constituent genes. Consequently, people with genetic differences can still have markedly similar phenotypes. Identical twins having identical training data can differ in their appearance. Parents of identical twins know how incorrect are the stereotypes of similar looks and likes. Wide differences in appearances of identical twins have been well documented. In an artificially intelligent system, such differences in models with same training data can emerge on account of stochastic nature of learning algorithms, evaluation procedure, and differences in underlying mathematical libraries. In life, different phenotypes are perhaps the outcome of interaction between the different test data, the environment as I have called it, faced by the model represented by the cells in your body. Training data is derived from historically significant learning events. Two people from different races can have more similar genotypes than two people from the same race, for example. However, people from the same race are expected to be phenotypically more similar, since phenotype is derived based on the test data, whose similarity is environment dependent. Also, phenotype is species independent. An eye cell for human is phenotypically more like an eye cell in the mouse than to a muscle cell in the same person, even though the eye cell in human and the eye cell in mouse would have different genotypes. To understand the learning model, let us work on establishing some equivalents with machine learning systems. You think about these equivalents in life for a moment before you show you some of our hypothesized answers. Learning algorithm in the human cell has to lead to greater order, since life leads to only one of the infinite possible outcomes, and hence perhaps the optimizer of life is entropy minimization. Evaluation procedure is not very clear. We are still building hypotheses around the same for our next meeting. Cellular machinery performs the task of underlying mathematical libraries. Epigenome determines the immediate response to the environment, which can be thought of as the test data. The way we assign functions to various parts of our DNA and non-coding RNA is to study them in a given context. However, most of the non-coded parts have context specific function, wherein context is extremely complex combination of different concentration of molecules within the cell, ultimately determined by environment around us and sequence in which the environment has unfolded over time. That is why we have struggled with assigning a function to large chunks of DNA. So, the summary of what all we have covered so far is well captured by the learning matrix, which with large number of constituent molecules and interdependencies can only be deciphered by application of machine learning tools to appropriately framed problems. During cell division, DNA is preserved. Even in sexual reproduction, DNA is mostly preserved. In a generation, very few base pairs change from parents to the offspring. Since we are yet to understand the evolution of the entire context in which the training data has been stored, we cannot yet classify any fragment of the training data as redundant. While formulating the model, our team was pleasantly surprised to discover plenty of emerging evidence supporting the learning model of life. We will cover two of the developments today that reinforce the learning model. One of the first examples we studied was of the synthetic cell created by John Craig Venter, a biotechnologist and a genetic researcher. Venter created the first synthetic cell in vitro, mimicking a natural cell for the first time. Venter and his team embarked on a mission to synthesize a minimal cell in silico, starting from a small bacterial cell. They selected a mycocyte bacterium cell with 1.21 million base pairs and identified the base pair coding for certain amino acids leading to specific proteins. He removed those genes for which proteins could be provided externally. He also removed most of the non-coding parts with no known function. The final cell had 483,000 base pairs, but the cell thus synthesized with these many base pairs was not viable. Attrition of apparently non-functional parts made life unviable. Venter's team then broke up the 1.21 million base pairs into 8 segments without any reference to the earlier failed experiment. On mixing and matching the 8 segments, a particular sequence of 531,000 base pairs was selected that formed a viable cell. This process highlighted the role of non-coding DNA in directing the expression of coding DNA and the presence of paired genes which were both indispensable despite essentially producing the same proteins. The viable cell thus formed had a cell division time of 3 hours compared to 1 hour of the normal mycocyte bacterium cell. Somewhere substantive learning had been lost in the transition from 1.21 million base pairs to about 500,000 base pairs. More importantly, however, the experiment revealed two paradoxical findings. Some of the genes which Venter thought essential had no impact even while they were knocked off. And second, some of the genes whose function could not be ascertained were critical for the cell to be viable. To remind you, this was a simple bacterial cell, and despite years of effort, resolving this paradox looked unfathomable. Venter's experiments on cell synthesis is a watershed moment, even though it is not treated as such. A study of the base pair patterns via machine learning can throw open the nature of learning at its very core to us. Another supporting evidence for existence of learning model emerges from the latest research on cell division. All of us have studied that mitosis is followed by equal division of the cell's content into two daughter cells that have identical genomes. In a 2019 study published in PLOS biology, the authors of the paper found that cell division is not identical in terms of DNA damage. The cell divides into a young daughter with lesser DNA damage and an old daughter with greater DNA damage. If you see the diagram, maximum DNA damage is capped at 63%, while the minimum damage progressively reduces at every stage, thereby preserving cellular immortality. Asymmetric cell division illustrates it's possible to reverse the damage or reverse the aging or reverse any disorder for that matter. The learning model of life represents a paradigm shift in the way we think about physiological sciences and has wide-ranging implications, some of which I will touch upon today. A major implication of the learning model is that every individual can prime their learning system by calibrating the environment around it. By calibrating your environment rightly, you become your rightest self. At EPIGENOME, our endeavor is to scientifically identify the right environment for you. To understand the implications further, we delve into our embryonic journey. The first stage of cell differentiation, embryonic stage, creates an environment gradient to allow for initiation of cell differentiation through ectoderm, mesoderm and endoderm. We have not been able to replicate the embryonic stage in vitro so far, as the environment gradient has not yet been understood for it to be created in vitro. Additionally, the connection with mother's circulatory system may be required for the learning to progress further. Looking from the lens of sexual reproduction, training data for the offspring is contributed by both biological mother and biological father. Energy metabolism related genes come from biological mother. In the case of conception via surrogate, the child will derive most of the learnings or epigenome from the surrogate mother, even though the training data has been mostly contributed by the biological father and the biological mother. There are markedly more dysfunctions in IVF, possibly due to exposure to hitherto not properly understood environment factors during the germination stage. These environmental factors could have led to new epigenetic markers in the embryonic stage and made the embryo more susceptible to dysfunction. Epigenome is influenced by biochemical cascades after birth. Phenotype modifications such as dark babies of white parents have been attributed to conjugal relationships several generations earlier. However, these are yet unproven, and while biological parentage of the dark baby has been determined, rare biochemical pathways that could have triggered dark babies with white parents in a generally white baby producing environment is yet undetermined. What has however been determined by closely controlled experiments is the impact of pup-licking and grooming on adult offspring. There was significantly higher pain thresholds and improved matrices for emotional health of the offspring if it was properly groomed during its childhood. The summary of all what we have covered so far is that the learning model provides a comprehensive framework to determine the optimum sequencing of the test data or the environment, as I put it, for desired life and health outcomes through right data sets and machine learning algorithms. The compute requirement of most such problems are pretty low and can be handled by routine AI systems prevalent now. While we have the means to change the training data, thanks to the recent advance in CRISPR-Cas9, the same is not at all required. What we have found is that changing the epigenome is good enough for us to build and maintain our health in the way we want to. And the tools to change our epigenome is through two biochemical pathways which contribute immensely to the learning, metabolic pathway and the signaling pathway. The pathways to learning, signaling pathway and metabolic pathway, which I spoke about earlier, have a close interlinking relationship. The generation of hormones is determined by availability of nutrient derived molecules in the body, which are a product of metabolic pathway, and the very presence of those hormones then determine the directionality of metabolic reactions in the body. A right pattern of molecules in the cells will promote the right learning by epigenomic changes. However, that pattern is complex enough to not to be understood so simply, as will become evident in the next few minutes. If we look at the energy metabolism in human cells, acetyl-CoA is the central molecule from all macronutrients including fats, proteins, carbohydrates. What causes the different response to different eating patterns then is not the macronutrients themselves, but the presence of hormones via signaling pathways, and the hormones are insulin, glucagon and IGF-1. Higher insulin levels suppress growth hormone and thereby IGF-1. Higher growth hormones will suppress insulin production. So, insulin and IGF usually have an inversely proportional presence in the cell. IGF-1 promotes phospholipids, cholesterol and amino acid synthesis, which can accelerate cell division and stop programmed cell death. Insulin promotes glycolysis and triglyceride synthesis required for triglyceride formation and fat storage without necessarily impacting rate of cell division. Glucagon promotes gluconeogenesis and ketogenesis in liver, leading to clearance of some of the accumulated fat in the liver cells. In simple words, insulin is fat building, IGF-1 is new cell building, that is growth promoting, while glucagon has a liver clearing effect. I specifically wanted to cover alcohol metabolism since many of you have alcohol every now and then. Most of the alcohol is metabolized in liver via acetaldehyde as an intermediate product. The end product acetate is harmless and can be a source of energy, but acetaldehyde is the problem child. Acetaldehyde increases the synthesis of the mature SREBP1 protein, which enhances fat accumulation in the liver. In your liver function test, it will reflect as an elevated aspartate transaminase or AST. Acetaldehyde further suppresses two hormones, adenopectin and PPAR alpha regulator of beta oxidation, thereby suppressing fatty acid metabolism. Together, these two effects promote a compounding effect leading to excessive fatty acid synthesis in the liver cells. If you have 30 ml alcohol, a small peg for example, or 1 pint of beer or 2 glasses of wine say, elevated acetaldehyde concentration remains elevated only for a small duration, about 1 to 1.5 hours. If however you have anything above 50 ml, damage can start accumulating by elevated acetaldehyde concentration for 2 to 6 hours. 120 ml of alcohol consumption may induce deposition of 10 grams fat in liver. Same continued for 21 days would cause an additional deposition of 210 grams liver fat, causing a 200 to 250% increase in fat deposition in the liver. Acetate, the final product of the alcohol metabolism, is distributed to whole body via blood. Chronic alcohol consumption may lead to brain using acetate as fuel source. Irrespective, acetate can convert to acetyl-CoA, the central molecule in energy metabolism which you saw earlier. Acetate is also produced in the gut by the gut microbe. We will talk more about gut microbiota in few minutes. There are some key messages from the assessment of toxic foods for other mammals, since we all share the similar learning lineage. Naked carbohydrate is bad even for herbivores. Even though left uncontrolled, they will gorge on it. Sugar is outright bad for cats, okay in small quantities for some species. Caffeine is generally bad but okay in small quantities for cows. Alcohol is generally bad but cows can tolerate it to some extent. Garlic and onion, for example, are bad for carnivores. Anything that has a degree of toxicity is metabolized in the liver. For instance, fructose from sugar, xylitol, caffeine, alcohol. Since liver weight is less than 3% of the body weight, such toxic compounds can be handled only up to 3% of daily macronutrient consumption, that is 2 tablespoons or 10 to 12 grams max in a day. And last but not the least, we share more learnings with herbivores than with carnivores. To understand the implications of metabolic pathways and hormone interlinkages, let us assess the generally followed diets from macronutrient perspective. If you have normal insulin sensitivity, you would be fairly okay with the generally recommended diet encompassing 300 grams carbohydrates, 150 grams proteins and 40 grams of fat. If you still go on a keto diet, you will have very low insulin levels, high IGF-1 levels and high glucagon levels, which means you can even indulge in some alcohol and some fructose abuse and get away with it, as intermittent gluconeogenesis and ketogenesis will clear your liver every now and then. If you are insulin resistant, however, the generally recommended diet will lead to constant triglyceride accumulation in your body, and liver as elevated insulin level will prevent gluconeogenesis and ketogenesis. Insulin resistance is caused by varying deficiencies or excesses of certain micronutrients and or heavy metals. So, the moot point is there are many layers of complexity in understanding what is suitable for whom. The only key message that I can deliver simplistically is that a minimum uptake of fats and proteins is required, since the body cannot make essential fatty acids and amino acids by itself, rendering the low fat diet a problem under any context. If you go on a low fat diet, whether it be high protein or high carb, universal effect is similar. That is, you will lose out on essential fatty acids, leading to high inflammation and oxidative stress in the body. And low fat diet will inhibit uptake of fat soluble vitamins A, D, E and K, causing several disorders including depression, irritability, lack of vitality, compromised immune system, frequent muscle or bone injuries, anemia and osteoporosis. Together, the above two factors will significantly increase the risk of cardiovascular disease, which ironically this diet was meant to minimize. One other factor to keep in mind is that sugar intake needs to be limited to 25 to 30 grams in a day, and if you consume alcohol and caffeine, perhaps better to eliminate it completely, as you would not want to overload your liver. At the same time, fiber is an essential carbohydrate whose intake needs to be a minimum of 30-35 grams, since it feeds your gut microbiota, whose role I will cover separately. Micronutrients play a catalyzing role in energy metabolism, either directly or via catalyzing hormone synthesis. For instance, water soluble vitamins, essentially B vitamins and vitamin C, iron and magnesium are directly involved in one or more of the reactions: glycolysis, Krebs cycle, beta oxidation and electron transport chain. These vitamins and minerals are also involved in synthesis of neuronal structures and neurotransmission, hence being critical for brain activity. Fat soluble vitamins exert their effects through various hormones and impact vision, cell differentiation, immune system modulation, as well as acting as antioxidants. Macro minerals play a significant role in metabolic processes. To maximize the benefits of fasting, it is recommended to have macro minerals even during period of no macronutrient uptake. One of the most underestimated macromineral is sulfur, which is a constituent of acetyl CoA. High sulfur food like broccoli, cabbage, cauliflower, a kind of medicine for people with multiple sclerosis and other autoimmune disorders. Phytochemicals are plant compounds which magnify the benefits of vitamins and minerals through yet to be discovered processes. In the initial studies with vitamins and mineral supplements, it was found that people who consumed enough vitamins and minerals through natural foods invariably gained more health benefits than those who relied purely on supplements. This led to the myth that natural compounds with same formula is better than synthesized one. However, With the discovery of phytonutrient, the conundrum stands resolved now. Okay, if a synthetic formulation can provide the vitamins, minerals and phytonutrients in the right proportion, there is no reason why it cannot foster the same benefits as a naturally occurring formulation in the form of a food item. There is an entire science behind the ability of certain nutrients to be extraordinarily beneficial in nano doses while being toxic even in relatively larger but still small doses. Some of the current trace minerals like iron started off as being toxic to early mammals and later became critical to certain life processes. Many such nutrients may yet not have been discovered. In fact, many of the trace minerals are on the borderline of micronutrients and nanonutrients. Hormesis is the name of the science behind such small exposures to certain nutrients being overall beneficial by priming the learning system. Homeopathy, a so-called pseudoscience, is based on sound principles of hormesis. However, the dilution levels, the determination of right allergens that needs to be administered to the patients is something that has not evolved in the last century on account of lack of credible research. The other pathway that determines who we are and our health outcomes exert its effects through various enzymes and hormones secreted either via ducts or directly in the blood by endocrine glands. The exocrine glands that secrete enzymes and hormones through ducts exert a direct localized effect in digestion, thermoregulation, lubrication and so on, while endocrine gland exert a more nuanced effect on the entire body by determining directionality of various metabolic pathways. At the same time, robustness of the endocrine gland system is influenced by several micronutrients we already covered. There is thus a circular relationship between metabolic and signaling pathways, which requires deep learning and complex pattern recognition to decipher. Micronutrient deficiency plays a major role in endocrine disruption. At the same time, exposure to many chemicals can also cause endocrine disruption. These chemicals appear everywhere in pesticides, plastics, cosmetics and even our food and water. If the goal of your life is to have right sleep, no unnecessary stress, well primed metabolism, less visceral fat, healthy muscle mass, then the hormonal balance that you should strive for is as follows. A. Low insulin, therefore high IGF-1. B. High oxytocin. C. Circadian monitored proper melatonin release. D. Optimum aldosterone. But the key question would remain how to achieve this balance. The balance may be achieved by correcting cellular composition of micronutrients and nanonutrients, having the right bacteria in our gut and triggering certain signaling pathways via high intensity interval training, fasting, whole body cryo, sauna and other such things. All this can be determined by studying the hormonal patterns across appropriately framed data sets. Gut microbiota is the final piece in our attempt to solve the jigsaw of pathways leading to learning. Gut microbiota play a key role in mechanisms affecting energy harvest from diet and energy storage in us. Microbiota promotes absorption of monosaccharides from the gut lumen with resulting induction of fat storage. Fasting induced adipocyte factor, a member of the angiopoietin like family of proteins, is selectively suppressed in the intestinal epithelium. If we were to become entirely sterile or germ free, our energy requirements would be higher by up to 12 to 15% and we would have limited ability to store fat. Some of the critical vitamins like B vitamins and vitamin K are produced by gut microbiota. Gut microbiota is one of the strongest modulator of the immune system, and a compromised gut microbiota can make us susceptible to frequent infections. People with impaired protein metabolism have also been demonstrated to have gained significantly from fecal transplant, thereby highlighting the important role played by gut microbiota in protein metabolism. Having understood that there are two main pathways to impact learning, the key question before us remains: has our learning been so far the status quo as of now, and what can we do to improve it in a meaningful way? To understand the learning path you have followed so far, we plan to utilize blood-based biomarkers which are a good representation of metabolic function and endocrine function. Our assessment of these would give us a good insight into the good, bad and the ugly part of your learning, also deriving insights on gut microbiota to an extent. Deeper tests may be required for some of you who have some parameters in the gray zone, but for a large segment we would be able to assess the status quo to a high degree of accuracy. The status quo is an impact of certain environmental factors. By tweaking the very same factors, we can move towards desired life outcomes. The extent of the tweak needs to be determined by recognizing patterns across the large data sets in an unbiased, impersonal manner, and the same can be done via application of machine learning. The endeavor would be to ultimately not restrict you to a narrow set of unpracticable approaches, but to find a set of activities and food items that you can continue to cherish while still working towards your desired health and life outcomes. What we need to understand is that the so-called universally health activities or foods is only a minuscule subset of available set of choices and is mostly impracticable and costly. However, the same objectives can be achieved with completely different activities and foods that fit your schedule and budget constraints much better.